அனைவருக்கும் வணக்கம். பயாலஜி தமிழ்நாடு சேனல் சார்பாக உங்களை அன்புடன் வரவேற்கிறோம். இன்றைக்கி நம்ம என்ன பார்க்க போகிறோம் அப்படின்னா, உங்களுடைய ஸ்டேட் போர்டு 12th ஸ்டாண்டர்ட் பயோ பாட்டனி புக்கில் இருக்கக்கூடிய 4th லெசன் பிரின்சிபிள்ஸ் ஆஃப் பயோடெக்னாலஜி அப்படிங்கிற ஒரு லெசனில் இருக்கக்கூடிய 1 வேர்ட்ஸ் கொஸ்டின்ஸ் அண்ட் ஆன்சர் பார்த்துட்ருக்கோம். அதில் பார்ட் 3ல் நாம் சந்திக்கிறோம். ரைட் த அட்வான்டேஜ் ஆஃப் எம்சிஎஸ். MCS facilitates the use of restriction enzyme of choice. MCS அப்படின்னா multiple cloning site. The foreign DNA fragment can be inserted into a plasmid with the use of restriction enzymes. Dash is a sequence from where replication starts. Origin of replication, ORI. Name the DNA segment that enable cells containing the vector to be selected. Selectable markers. Name the site in which one or more restriction enzyme used during cloning. Cleavage site or restriction site. Dash helps in identifying and eliminating non-transformants. Selectable marker. Name the site of vector in which alien DNA is linked. Recognition sites or cloning sites. Dash are extrachromosomal self-replicating double-stranded circular DNA molecules found in bacteria. Plasmids. Which type of plasmid is a reconstructed plasmid and most widely used as cloning vector? PBr322. How many base pairs are present in PBr322? 4,361 base pairs. In PBr22, P denotes plasmid. In PBr322, B and R represent Bolivar and Rodriguez. In PBr322, what is 322? It is the number of plasmid developed in the laboratory. What is AMPR? Ampicillin resistant gene. What is TATR? Tetracycline resistant. What is rope? Replication proteins. Ti plasmid is found in Agrobacterium tumefaciens bacteria. Which bacteria are responsible for inducing tumors in several dicot plants? Agrobacterium tumefaciens. Name the gene present in Ti plasmid to transfer tDNA from one bacterium to other bacteria or plant cell. TRA genes. What is onc gene? Genes responsible for oncogenicity. Which bacterial plasmids have used for introduction of genes of desirable traits into plants? Agrobacterium plasmids. Propagation of the recombinants and molecules must occur inside a living system or a host. What are the host cells available for gene cloning? E. coli, yeast, animal or plant cells. E. coli is the most widely used for cloning experiment. Why? Genetic makeup extensively studied, easy to handle, can accept a range of vectors, studied for safety. Cells divide within 20 minutes. Name the genetic material which is hydrophilic and cannot pass through cell membranes. DNA. How is a bacterial cell made competent to take up DNA? By using divalent cation such as calcium. For the expression of eukaryotic proteins, dash cells are preferred. Eukaryotic cells. What are the two kinds of gene transfer methods in plants? Direct or vectorless gene transfer and indirect or vector mediated gene transfer. In which method, gene transfer methods, the foreign gene of interest is delivered into the host plant without the help of a vector. Direct gene transfer method. Name the chemicals induce DNA uptake into plant protoplasts. Polyethylene glycol and dextran sulfate. In which method the DNA is directly injected into the nucleus using fine tipped glass needle or micropipette. Microinjection. In which technique the protoplasts are immobilized on a solid support. Agarose on a microscopic slide. In which method of gene transfer a pulse of high voltage is applied to protoplasts to form transient pores in the plasma membrane. Electroporation. Dash or the artificial phospholipid vesicles are useful in gene transfer. Liposomes. Which type of vectorless gene transfer is advantageous? Liposome mediated gene transfer. Liposome and tonoplast of vacuole fusion resulted in gene transfer. This process is called liposuction. In which technique foreign DNA is coated onto the surface of minute gold or tungsten particles and bombarded onto the target tissue. Biolistics or gene gun or microprojectile or shotgun method. Indirect method of gene transfer is otherwise called vector mediated gene transfer. Gene transfer is mediated with the help of a plasmid vector is known as indirect or vector mediated gene transfer. Which vector is used for plant transformation mostly? TI plasmid from Agrobacterium tumefaciens. Abbreviate TI. Tumor inducing. What is tDNA? Transfer DNA, which is known as the natural genetic engineer of plants. Agrobacterium tumefaciens. Name the gene confers resistance to antibiotic kanamycin. NPT2. After the introduction of rDNA into suitable host cell, It is essential to identify those cells which have received the rDNA molecule. This method is called screening of recombinants. Which method of screening is a powerful method used for screening of recombinant plasmids? Insertional inactivation or blue-white colony selection method. The gene LAC-Z encodes the enzyme beta-galactosidase. Chemical name of X-gal. Fibromo 4-chloro-indolyl beta-D-galactopyranoside. Dash is a gene that produces a protein that provides cells with resistance to antibiotic. Antibiotic resistance marker. In which technique the pattern of colonies growing on a culture plate is copied? Replica plating technique. In which technique used to separate different biomolecules with positive and negative charges? Gel electrophoresis. In which technique application of electricity leads to the migration of molecules according to the type of charges they have? Agarose gel electrophoresis. மேலும் பல தகவல்களை தெரிந்து கொள்வதற்கு பயாலஜி தமிழ்நாடு சேனலை சப்ஸ்கிரைப் பண்ணுங்கள். நன்றி, வணக்கம்.
பலருக்கு சேவை செஞ்ச நீயும் ஒரு ஹீரோடா. ஒரு கைப்பிடி சோறானாலும் ஷேர் செஞ்சு பாருடா.